I'm Dr. Fakir Ravani, and this is clinical microbiology, susceptibility testing, and systematic approach to infectious diseases. Here's an overview of the human immune system. We can think of the immune system as innate immunity and adaptive immunity. The components of innate immunity include physical and chemical barriers. So physical, you can think of the skin, which basically prevents pathogens from entering the body. And chemical barriers, you can think of components of the saliva, and perhaps most importantly, the gastric acids that basically kill pathogens. Other components of innate immunity include mucus, the complement system, and innate cells such as macrophages, dendritic cells, granulocytes, natural killer cells. In general, the innate immunity is non-specific or broadly specific molecular patterns. And the actual immune response is through cytokines and chemokines, which lead to inflammation, phagocytosis processes, the complement activation, as well as activation of effector cells. Now, innate immunity is extremely important because in healthy individuals with an intact immune system, many features constitutively are present. In other words, you don't really need prior infection or immunization in order to have innate immunity. And the response is immediate. So it takes typically minutes to hours for the innate immunity to kick in and fight infection. However, it only persists for a few days, so it doesn't really give you long-term protection or memory. That's where adaptive immunity comes in. So the components of adaptive immunity includes humoral immunity, such as B lymphocyte cells and antibodies, as well as cell-mediated immunity, such as T lymphocytes or T cells. So adaptive immunity is antigen specific to a given pathogen. So it's not very broad, it's very specific to specific infectious diseases. And the actual response is through activation, proliferation, and differentiation of antigen specific B and T lymphocytes. It involves secretion of antibodies that can fight pathogens, as well as effector activity of T lymphocytes, and of course generation of memory cells, which lead to long-term immunity. And these mechanisms get activated either through actual natural infection or through immunization. Now, the first time the body is exposed to a pathogen, the adaptive immunity will take several days to develop, and that's referred to as primary response. So primary is the first time the body sees that pathogen. However, once the immune system has adapted to that pathogen, the next time the body encounters that same pathogen, there will be a rapid response due to the memory that develops. So because of that, there is a long-term duration of adaptive immunity. Now, once the adaptive immunity clears an infection, there is a contraction after elimination of pathogen that typically happens in a few weeks after the infection is cleared. And what contraction means is essentially, through adaptive immunity, the body will develop a lot of antibodies. And then once the infection is cleared, these antibodies start to wear off. So the levels of antibodies after a few weeks go down to very low levels, and that's called contraction. However, because the memory cells remain, and there are memory B cells are the cells that make antibodies. So memory B cells will last months and years, and there are also memory T cells that will probably last for the entire lifetime. And so the next time the body is exposed to the pathogen, because of this long-term memory, the body is able to expand the antibody levels very quickly. Now let's take a look at the hematopoiesis process. We're not going to go into too much details on this. We just need to know what the end products are and what their roles are in the immune system. Of course, this all starts with the hematopoietic stem cells, and these stem cells essentially differentiate into different cells, and these are the cells that exist in the blood. So, in general, we have three different kinds of cells in blood. Of course, we have red blood cells. Red blood cells, another name for it is erythrocytes. We have platelets. Another name for platelets is thrombocytes. And then everything else that you see here are essentially white blood cells. And another word for white blood cells are leukocytes. So now, because white blood cells have an important role in fighting infections, we're going to go into more details of the different types of cells that constitute leukocytes. Essentially, anything on the right-hand side on this pathway are lymphocytes. So we have B lymphocytes, we have T lymphocytes, and we have natural killer cells. And then everything to the left essentially comes from myeloid progenitor. So these are basophils, eosinophils, neutrophils, mast cells, and monocytes. Now, when we measure white blood cells in the blood, we typically count how many cells are in a microliter of blood. So, the normal range is anywhere between 4,000 to 11,000 cells per microliter. Now, if somebody has low levels of white blood cell, that's referred to as leukopenia. And of course, the other end is if somebody has too much white blood cell, it's referred to as leukocytosis. And that typically happens when people have an infection. Not every infection will lead to increased white blood cell, but if it is a type of infection that requires white blood cells to fight it, the body will start making more white blood cells. So, a typical sign of an infection would be if somebody has elevated white blood cell or, more appropriately called leukocytosis. And that would be if somebody has a level 12,000 or higher. Now, here on the right-hand side, you can see the shorthand way of writing some of these lab values. So on the left hand side is white blood cell, on the right hand side is platelet, at the top is hemoglobin, and at the bottom is hematocrit. And this is a shorthand when typically clinicians, when they work up patients, this is how they write. So you don't have to write white blood cell, you just write numbers here instead of these labels. And typically, if somebody says white blood cell is 4, they really mean 4,000. They're not going to write 4,000 or 5,000. So they typically write, like 6.2 or, 13. So if somebody writes 13 here, they really mean 13,000. Now, if you know somebody has leukopenia and they write 50 here, they're really talking about 50 cells. They're not going to write 0.050. So you just have to use it in the context. The same with platelets. So, typically people ignore the 3 zeros at the end. Now, hemoglobin and hematocrits are more straightforward, so the numbers are not too large. They're not in the thousands. So when somebody writes here hemoglobin is 14, they actually mean 14. And hematocrit is in percentage, so that one is easy. Now let's take a look at the 6 basic types of leukocytes. So eosinophils constitute about 1 to 3% of all white blood cells. Basophils and mast cells, less than 1% of white blood cells. Neutrophils are extremely important. Neutrophils are the primary mechanism of fighting bacterial infection. So when it comes to bacteria, neutrophils have a primary role. And that's why you will see a large portion of white blood cells, typically 50 to 70% are neutrophils. Now, an important concept when it comes to neutrophil is the concept of segs and bands. So what we mean by segs or PMNs or polymorphonuclear leukocytes or just polys is that these are essentially mature neutrophils. And then the immature neutrophils are called bands, so band cells. And essentially, when the body makes new neutrophils, initially they are bands, and then it takes time for them to mature and become segs. So normally, most of the neutrophils circulating in blood are mature in the mature form, with the nucleus of the cell being divided or segmented. So that's where the name segs comes from. Now, during infection, because the body tries to make more neutrophils than usual, you will see an increase in the band or immature neutrophils, and that's typically referred to as bandemia or left shift. So left shift meaning the cells have shifted to the immature side. And so if you see more than 10% of the neutrophils in the band cell, that's typically called bandemia. So this percentage here is 3 to 5% of white blood cells are normally bands, and white blood cells, about half of them, 50 to 70% are white blood cells. So this 3 to 5%, you can think of it as typically 6 to 10% of neutrophils. So if you see more than 10% of neutrophils as bands, that's typically clinically significant. Now, other types of leukocytes include monocytes, and they actually progress to become macrophages, and those are 1 to 6% of white blood cells. And then we have lymphocytes and their derivative plasma cells, and a big portion of white blood cells are lymphocytes, and they have an important role in developing memory. But also they have a big role when it comes to viral infections, and of course they also important in other types of infections, including bacterial and fungal infections. And lastly, we have dendritic cells, less than 1% of white blood cells. Now here are some more details about the function of each leukocyte type. In general, eosinophils have a role in allergic reactions, including allergic reaction to antimicrobial agents. They also have a role in fighting parasitic infections. Basophils and mast cells are also active during allergic response, especially anything with histamine release, and they have a role in inflammation. As I mentioned, neutrophils have a big role in bacterial infection, but they're also important in fungal infections. Macrophages are not only important in bacterial infections, but they're also antigen presenting cells, so they activate lymphocytes. And of course, the three types of lymphocytes. B lymphocytes are the ones that result in plasma cells that make antibodies. B lymphocytes themselves are also antigen presenting cells, so they can work with T cells. T cells, there are different types. There are CD4, CD8 that are most commonly important types of T lymphocytes. They are not only important in developing long-lasting memory to bacterial infections, but they also have an important role in fighting viral infections. We have natural killer cells, and lastly, we have dendritic cells that are also antigen-presenting cells. Please pause this video and review the function of each of these leukocyte types. Now that we have a good foundation, let's take a look at our first learning objective. Given the results of a specimen stain, list the most likely organisms present in the specimen. And the specimen would be from the site of infection. So this could be blood. If somebody has infection in the blood, referred to as bacteremia, or could be urine or sputum or CSF or other types of specimen. Let's take a look at different types of bacteria. So in general, bacteria are divided into different groups based on how the cell wall is stained. So if you look at bacteria under microscope, you're not going to see anything because they're transparent. So in order to see them under microscope, we have to use some techniques to stain them so we can see them. If we look at bacterial cells, you will see inside the cell, there is a genetic material, and then the cell is surrounded by a cytoplasmic membrane or the cell membrane. And then what you will see outside of the cell membrane is a cell wall, which is essentially made of peptidoglycans for most of these bacteria. And that's what we stain. So we stain the cell wall, and the cell wall is what gives the shape of the bacteria. So they will have different shapes. So, either rod shaped or spiral or other shapes that we will talk about shortly. And one stain that we can use is called the Gram stain. So Gram was, is the name of the person who came up with the technique. And some of these bacteria, when you use the Gram stain, they will turn blue, and we call that Gram positive. So something that's particular about Gram positive is that they have the cell membrane, and then outside of the cell membrane, they have a thick layer of peptidoglycan, and then this layer gets, this is the cell wall that gets stained blue. And then we have another group of bacteria that are called Gram negative. These essentially stain, pink using Gram staining. And something that's particular about the Gram negative is that, they still have the cell membrane followed by a thin layer of peptidoglycan. And the reason this is thin is that on top of the peptidoglycan, there's a second outer membrane. So there are two membranes. So that's a major difference between Gram positive and Gram negative, that, Gram negatives have this outer membrane. So the peptidoglycan is thinner. And then we have a third group that just doesn't get stained using the Gram technique. So then we will have to use a different type of staining. So the acid-fast staining is used. A good example is mycobacteria. For example, if you think of tuberculosis. So these get stained with acid-fast. And then the fourth group is what we call atypical, because they do not have cell wall. So, they do not have peptidoglycan, because these three have peptidoglycan that can get, one way or another, they're going to get stained. But the atypicals, because they don't have a cell wall, they don't get stained. So, any of these techniques you use, they will not be stained. So these are atypical. So these are in general the four types of bacteria that are clinically important. Now let's take a look at some of these staining and microscopy techniques. So as I mentioned, the Gram stain is the most commonly used technique, and this technique can stain bacteria, but also some fungi species can also get stained. Certain bacteria, most importantly mycobacteria, do not get stained using Gram stain, so acid-fast is what we use. And then there are specific techniques that are used when it comes to different types of fungal species. Let's take a look at taxonomy of bacteria. So taxonomy is essentially the classification of organisms in an ordered system that indicates a natural relationship. You may be familiar with Linnaean taxonomy, so that's the name of the person who came up with this system, which ranks organisms by domain, kingdom, phylum, class, order, family, genus, species, and subtype of species for some species. For example, E. coli, Escherichia coli, is classified here, and the most useful in general is the family, genus, and species. And occasionally, for example, when it comes to E. coli, it is useful to know the order as well. And the reason is there are a few clinically important organisms that belong to the family of Enterobacteriaceae, such as E. coli. And recently, some of them were reclassified to different families. So we used to refer to these by the family Enterobacteriaceae. And because some of them are no longer in this family, it is clinically useful to refer to them by the order Enterobacterialis. Now, a strain is a subgroup within the species that has unique characteristics that distinguishes it from other strains. For example, E. coli is the species, and E. coli O157H7 is one of the subtypes of E. coli, and that itself can have different strains. For example, these strains are identified by a sequence number. So that's an example of strains. Now let's take a look at some specific examples of Gram-positive bacteria. Now in general, whether it's Gram-positive or Gram-negative, these bacteria can be either aerobic or anaerobic. So essentially aerobic means that they require oxygen for replication, Anaerobic means that they can replicate in the absence of oxygen. And these definitions get more nuanced from a microbiology standpoint, but for us, clinically, that will suffice to distinguish between aerobic and anaerobic. And then with gram-positive specifically, we can look at morphology. So morphology is basically the shape of these and how they group together when you look at them under the microscope. So essentially, we can see cocci, and this is essentially in circular shape, and they can be in clusters. So cluster means that several of them are clustering together. And then you can have cocci, which is essentially circles in pairs or chains. So pairs will be a couple of them, and sometimes that's referred to as diplococci. And then, if you have a chain, they don't cluster. It's just, just like a chain. And then we have rod shape that are called bacillus, and the plural for it would be bacilli. And I should say cocci is a plural. The singular form is coccus. So if you're talking about the single bacterium, it will be a coccus. And then so for gram-positives that look like cocci in clusters, typically there's a test they do, then these tests catalase positive. And then on top of that, in the microlab, they can do a coagulase test. And if you have coagulase positive, then you have a very important, clinically important organism, Staphylococcus aureus. Okay, and the reason we're talking about this is because it takes some time to identify what the organism is. And if somebody has an infection and we don't know what it is yet, but they look at it under the microscope and they say, well, it looks like cocci in clusters, and we also did a coagulase test and coagulase test was positive, as clinicians we can anticipate Staphylococcus aureus, so we can give appropriate treatment. So that's why we're covering this. We're not trying to become microbiologists, but as clinicians you can use this clinically to help patients. And if they say the coagulase is negative, you have several options, and together all of these are referred to as coagulase negative staph or coagulase negative staphylococcus. And there are several species that are coag negative, and I have listed 4 that are more common. And Staphylococcus lugdunensis is one that can cause severe infection, so that's why I have bolded it. But so these are typically present on the skin. The same with Staphylococcus aureus. All staphylococci are typically present on skin, and occasionally they find their way into the body and cause infection. And then for the catalase negative group, which they do not look like a cluster, so they will be in chain or maybe pairs. Then we can do the hemolysis test. So what a hemolysis test would be is that essentially an agar is essentially a plate. So what we do is that we use sheep blood, and we basically let these organisms grow and see what they do to the blood cells. So the beta hemolytic group, which you can see in this picture, essentially they lyse blood cells, so they will break it down and it will basically clear the blood, and that's called beta hemolytic strep. And the alpha essentially reduce hemoglobin, and they cause this greenish color. And then there is a third group that don't really do anything to the blood, and these are referred to as non-hemolytic because they didn't break it down. Essentially that's how we group these. So beta hemolytic strep, alpha hemolytic strep, and non-hemolytic. So in the beta hemolytic group, we have 2 important Streptococcus species. So Strep pyogenes, which is sometimes referred to as group A strep. And then we have Streptococcus agalactiae, which is referred to as group B strep. So these are important, and you should know these interchangeably, because sometimes people may refer to them as group B strep. So they might say, okay, for this patient, we are trying to cover group B strep. So you should know that they are referring to Streptococcus agalactiae. And if they say we want to cover group A strep, you should know they're talking about Streptococcus pyogenes. And then for the alpha hemolytic group, the most important is Streptococcus pneumoniae. This is very important because it causes respiratory infections, and we'll cover this in detail throughout the course. And then there's also the viridans group Streptococci. When it says group, there are multiple species, and there's really no need to know specific species. They're collectively referred to as viridans group Streptococci. And then the non-hemolytic, we have two Enterococcus. In the old days, these were also called Streptococcus, but nomenclature has changed over time. So nowadays, these are called Enterococcus, but they're also in the same group as the rest of the strep. Enterococcus faecalis and Enterococcus faecium, these are two organisms that are kind of the frenemy, so they sometimes they are good because they can be as part of the flora in the body and do some good, and sometimes they can be bad and they can cause an infection. So that's why they're tricky. So you have, and we'll cover this throughout the course. So sometimes, for example, they might be in the urine and prevent urinary tract infection. Other times they might be the cause of urinary infection. So it becomes tricky when we identify this in a specimen. So a lot of caution should be used. And then lastly, these are just the gram-positive bacilli, so Listeria, Bacillus anthracis, and Corynebacterium diphtheria. Now let's take a look at gram-negative bacteria. Again, we're focusing on aerobic for now. So you will see that majority of gram-negative are actually rod-shaped. So these are bacilli. And what we do with gram-negatives is that we do a lactose fermenting test. So if they can ferment lactose, we call them, actually the important thing is to identify what's not fermenting lactose, and they're typically referred to as non-fermenters, okay? But if they do ferment lactose, then, they do oxidase tests, and, for this course, you don't have to worry about the oxidase test, but know that when it's lactose fermenting, for the most part, we're talking about Enterobacterialis, and Enterobacterialis is a family of several organisms, and Enterobacterialis used to be called Enterobacteriaceae. And the important organisms in Enterobacterialis family is Escherichia coli or E. coli, Klebsiella species. So when it says SPP, that means there are multiple. So for example, there is Klebsiella pneumoniae and there is Klebsiella oxytoca. And there is Enterobacter cloaceae. There are several Citrobacter species. There are Serratia species, Providentia species, and Protease species. So these are all under Enterobacterialis. And then we have, other lactose fermenters, so Aeromonas, Pasturella, and Vibrio species. And then when it comes to non-lactose fermenters, and this is bolder because these are very important organisms because they typically are very resistant to a lot of antibiotics. So the most important would be Pseudomonas aeruginosa. So this causes a lot of severe infections and becomes difficult to treat. And also Acinetobacter baumannii, which is less severe than Pseudomonas but can be more resistant to a lot of organisms. And then, Stenotrophomonas and Burkholderia also can be very, very resistant. And this typically, Stenotrophomonas and Burkholderia typically more common in immunocompromised patients. And then for the cocci in the gram-negative group, we have Neisseria meningitidis, Neisseria gonorrhoeae, and then we have Haemophilus influenzae and Moraxella catarrhalis. These two are actually kind of between cocci and bacilli, so they're referred to as coccobacilli. So the shape under microscope is between a circle and a rod. And then spirochetes, which are essentially look like a spring. So we have a couple of them, Troponema pallidum, as well as Borrelia burgdorferi. Now let's take a look at some anaerobes. So anaerobes, gram-positive is Peptostreptococcus, and then we have a series of Clostridium and Clostridioides difficile. So Clostridioides difficile also used to be called Clostridium difficile, and nomenclature changed a few years ago. And we will cover these later in the course. And the gram-negative anaerobes, essentially the most important you need to know is Bacteroides, because any intra-abdominal infection, Bacteroides have an important role. Atypical bacteria include chlamydia species, including chlamydia pneumoniae. That's not a sexually transmitted infection. It can cause, pneumonia. And then we have the sexually transmitted chlamydia. Also, we have Mycoplasma and Legionella. And then with the acid fast, we have Nocardia species, we have Mycobacteria, as well as Actinomyces.